So today I'm going to be presenting my second genetics exam mini video. I did prompt 2, which is reverse mapping. So here's our genetic map. It shows 3 linked loci, all with separate alleles. We have our coefficient of coincidence, which is 0.75. Our R1 value is going to be 18 centimorgans, and our R2 value is going to be 12 centimorgans. So in certain species of turtles, a dark green color is dominant to a pale green color, a plain shell is dominant to a spotted shell, and normal flippers are dominant to gray-tipped flippers. So down here I put the dominant and recessive traits and their alleles just for clarification. So in this cross we have a dark green spotted shell normal flippered turtle and a pale green plain shell gray-tipped flippered turtle. When we cross these two turtles, we get a F1 generation that is heterozygous for all traits. So we take this heterozygous individual, we see that it matches our genetic map, and we test cross it with a homozygous recessive individual who is pale green, has a spotted shell, and has gray-tipped flippers. So this test cross, it gives us 8 different gamete types. I have grouped the gamete types with what event would cause them to occur. So these two would take place in a double crossover event. These two could take place in a single crossover event in the R1 region. These two could take place in a single crossover event in the R2 region, and these two would take place when no crossover event occurs, so these are the parental type gametes. So in this column, I've calculated the probability of each event occurring. So if you want to calculate the probability of an offspring with a specific genotype of little g, little p, little f, you would go to a chart, you would see that it occurs as a double crossover event. So this probability I've calculated right here gives us the probability of any double crossover event occurring. As you can see, this is the equation I used. Then I plugged in my values and I get the 1.62%. However, to find the probability of a specific DCO gamete, you would take 1/2 of that percent. So when I do that, I get 0.81% chance of there being a little g, little p, little f gamete. So now if you want to calculate the probability of an offspring with the specific genotype big G, big P, little f, you'd once again go see that that would occur as a single crossover event in the R1 region. So to get the 6.38%, I used this equation, plugged in R values, but to find the probability of a specific single crossover event, you would do 1/2 of that value. So I do 1/2 of that percentage and I get 8.19%. So that tells you that there is an 8.19% chance of a gamete with the genotype big G, big P, little f. Here's my references, and thank you for watching.